Hello everyone, this is SHARMIA. In this video, we are going to see about what is meant by irreversible cell injury. First of all, what is meant by cell injury means cell injury occurs when cells are exposed to harmful stimuli, which disturb their normal structure and function. Harmful stimuli includes that may be genetic or acquired causes. Genetic means in Down syndrome, sickle cell anemia. These are the harmful stimuli. Due to that, cell will form as an abnormal substance. In acquired causes, there will be hypoxia, ischemia, reduced oxygen and blood supply, physical trauma like accidents. In these conditions and all, cell will have some damage. This is called cell injury. Irreversible cell injury means if the injury stimuli is for more period of time, the duration of the time is increased, or the injury is severe, means in this condition the cell will die, undergo necrosis or apoptosis and die. It cannot survive after the harmful stimuli is removed. Even after the harmful stimuli is removed, the cell cannot survive. The cell undergo death. This event is called irreversible cell injury. There are two types. One is necrosis and other one is apoptosis. Necrosis means mostly result of the denaturation of the intracellular proteins and the enzymatic digestion of the lethally injured cell. Means denaturation. The proteins, enzymes are all digested and the cells will lose their membrane integrity and all the intracellular contents will leak out. The enzymes mostly lysosomes will present in each cells. The lysosomes will digest the cells. Necrosis means uncontrolled death of the cells, which we cannot control. There will be continuous death of this cells. Nuclear changes includes necrotic cells, increased eosinophilia count. In microscopic, we can identify through the pink appearance of the cells. Nuclear changes will appear in three patterns due to the breakdown of the DNA. First one is karyolysis. Karyolysis, the DNA will get lost and base of taken the chromatin fade away. Pignosis means the nucleus will get string. First one, there will be inside the nucleus. Karyolysis, there inside the nucleus the DNA will get lost. In pignosis, the DNA will get stringed. In karyolysis, the nucleus get a fragment broken down and disappearance. The nucleus will dissolve and there will be a disappearance of the nucleus. First, karyolysis, in which there will be loss of DNA. In pignosis, the nucleus get stringed, and in karyolysis, the nucleus will fragment and disappear. Type of necrosis, coagulative necrosis. It is a form of necrosis in which the architecture of the dead tissue is preserved due to the denaturation of the structural proteins and enzymes. Example, necrosis affecting the heart, kidney and lungs. Next one is liquefactive necrosis. In this, digestion of the dead cells results from the formation of the liquid viscous mass called pus. Example, abscess, boil, necrosis of the brain. Next one, and gangrenous necrosis. It's clinical. In this, there will be the necrosis of the limb, especially the lower limb. It will get loss of the blood supply and undergo necrosis. There are two types of necrosis. One is wet. Wet means when a bacterial infection is superimposed, there will be a more liquefaction and which will lead to a necrosis in the limbs. Second one is gaseous necrosis. It's a special type of the coagulation necrosis which occurs in the condition tuberculosis. In this, cheese-like, more fragile white appearance of the area where necrosis happens. Next one, types of necrosis. Fat necrosis refers to the focal area of the fat destruction due to the release of activated pancreatic lipases into the pancreas and peritoneal cavity. Fat necrosis is of two types: enzymatic fat necrosis, which occurs in the pancreas; traumatic fat necrosis, which occurs in breast. Second type is fibrinoid necrosis. It's a form of necrosis which is seen in the immune reactions involving blood cells. These are about necrosis. Necrosis means uncontrolled cell death. Apoptosis. Apoptosis means programmed cell death. Is generally regulated cell death induced by regulated suicidal program by injured cell. It is referred to as a programmed cell death. Apoptosis is a pathway of cell death in which cells will die due to the activating enzymes that degrade its nucleus and its DNA, and next to the nucleus and next to the cytoplasmic proteins. What are the causes of apoptosis? Physical destruction of embryos. Epithelial cell will get lost in intestinal epithelium. Death of the cells such as neutrophils will get lost, and regression of the lacting breast. After lactinating period, the lactinating breast will regret. And apoptosis in pathological condition, radiations due to chemotherapy drugs, cytotoxic drugs, hypoxia, degrees toxic supply. This will damage the DNA. The DNA will not get repaired in irreversible cell injury. It will undergo apoptosis, a programmed cell death. Gene-related diseases of the central nervous system, cell death and viral infections. These are some of the examples. Morphological changes in apoptosis, programmed cell death includes cell will get stringed, chromatin will get condensed inside the nucleus, formation of the apoptotic bodies, phagocytosis of the apoptotic cells or cell bodies, usually macrophages. Mechanism of the apoptosis. Apoptosis which result from the activation of the enzyme called caspases. Caspases will exist in an inactive proenzyme within the cells only, and it will progress of apoptosis will divided into initiation. Caspases become catalytically active, and execution phase. In execution phase, degradation of the cellular components will occur. Next, activation of the caspases depends upon the balance between the pro-apoptotic and anti-apoptotic proteins. Two pathways there are in caspase activation. First one is mitochondrial pathway, and next one is the death receptor pathway. Difference between what are the difference between necrosis and apoptosis? In necrosis, the cell will become enlarged. Apoptosis, the cell will get shrinked. Necrosis happens as pycnosis, karyolysis, and karyolysis. Pycnosis means loss of the DNA molecule. Then only it will get stringed and dissolved. In their fragments, it will break down. Here, plasma membrane get disrupted. They are intact. Cellular contents will enzymatically digested and leak out of the cell. There, it will be released in apoptotic bodies. Here, adjacent inflammation may happen frequently. There will be no inflammation. Here, there a pathological role will take place. That with changes, macroscopical changes will get seen. There will be physiological and also pathological. Mechanisms of cell injury. First one is depletion of the ATP. First, in hypoxia and ischemic condition, there will be first depletion in the ATP. Mitochondria will not get perfect oxygen supply. Without oxygen supply, it cannot generate ATP. Mitochondria get damaged. Second, mitochondria will get damaged. How mitochondria will get damaged means there will be increased input of the calcium. This calcium will accumulate in the mitochondria, and the mitochondrial swelling will happen. Next one is the influx of the calcium, and calcium homeostasis will get lost. Next one, accumulation of the oxygen free radicals. How oxygen free radicals formed means it is formed in the mitochondria. In mitochondria, nutrients will converted into electrons by using an electron, oxygen, and glucose. It will convert. By converting the oxygen, electrons, and water H2, it will convert oxygen into water and excrete in mitochondria. But in oxygen depleted condition, membrane damage, mitochondrial damage condition, this electrons will leak out, and one electron will combine with oxygen only separately, so that it will form an oxygen free radicals. Oxygen free radicals will further damage the mitochondria, DNA, proteins, ribosome synthesis, etc. Next, defects in the membrane permeability. Permeability will get lost. Due to permeability loss, the influx of the sodium, potassium will get collapsed, and leads to more accumulation of the sodium, and leading to influx of water. Due to more water influx, it will cause the cell swelling. Then damage to the DNA and proteins will get damaged due to the formation of oxygen free radicals. Mechanisms of cell injury is decreased ATP will lead to downstream effects. Mitochondria will get damaged, which lead them pro-apoptotic proteins. End of calcium will increase the mitochondrial permeability, and activation of the multiple cellular enzymes, which leads to mitochondrial damage, and more accumulation of the calcium inside the mitochondria, and mitochondria get swollen. Next, increased reactive free radical. It will further damage the membrane, proteins, DNA, RNA, etc. Next one is membrane damage. There are two types of membrane: plasma membrane, lysosomal enzyme membrane. First, plasma membrane is the outer layer of the cell. In that, loss of the cellular components, because without membrane, if the membrane gets lost, means intracellular compounds will leak out. In lysosomal membrane damage, enzymatic digestion of the cellular components. Lysosome's main role is digesting the cellular components if the cell get damaged. So that role will be taken by the lysosomes. Next one, protein misfolding, DNA damage. This will activate the pro-apoptotic proteins. It will digest the cell. It's a program to cell death will occur. As ATP depletion will reduce the ATP, and ATP synthesis will get reduced in most conditions due to hypoxia, which can lead to cell death. The major causes of the ATP depletions are reduced supply of the oxygen and nutrition. Then mitochondrial damage. Mitochondrial damage and actions of some toxins like cyanide. Next one is influx of the calcium and loss of calcium homeostasis. Calcium ions are important mediators of cell injury, because low level of the intracellular calcium will protect the cells from injury. Yes, intracellular calcium level must be low, because it only protects the cells from injury. But in these conditions, ischemia and skeleton toxins cause an increase in the intracellular calcium level, which will initially, due to more increase in the intracellular calcium, it will accumulate in the mitochondria only. Mitochondria will get swollen and damaged. How influx of the calcium and increased intracellular calcium will cause cell injury. How which mechanism it follows means the accumulation of the calcium in the mitochondria will result in the opening of the mitochondrial permeability and transition pore. Increased cytosolic calcium will activate the number of lysosomal enzymes. If there is more increase in the calcium, means it will activate the lysosomal cells. Lysosomal cells start to digest the cellular contents. Increased intracellular calcium causes the activation of caspase. Yes. In this calcium injury, in cell injury conditions, extracellular calcium will enter into the intracellularly, and then it will increase the cytosolic calcium getting increased by accumulating in the mitochondria and the plasmic reticulum. It will activate the ATPase, decrease ATP, phospholipase, decrease phospholipids, and the membrane get damaged. It will activate the protease, disruption of the cytoskeletal proteins will further damage the membrane. Endonucleus will get activated, then nucleus will get damaged. So calcium play the major role. It will activate everything, and every intracellular substances will get damaged. First, mitochondria, ATP production reduced, phospholipids, so cell membrane get interrupted, proteases, proteins get interrupted, endonucleus, nucleus get interrupted. Next, accumulation of the oxygen free radical. Cell injury is induced by the free radical causes cell damage in many pathological conditions, such as chemical, radiation injury, ischemic repercussion injury, cellular aging, and microbial killing by phagocytes. Defects in the membrane permeability. Membrane damage of cell will cause cell injury and cell death. Most important sites of the membrane damage is mitochondrial membrane, plasma membrane, membranes of lysosomes. First, necroptosis. Necroptosis is a form of cell death that shares both necrosis and apoptosis. Morphologically, it resembles necrosis, but in some mechanism only it will follow apoptosis. Mostly uncontrolled cell death, only necroptosis follows, but in some conditions apoptosis also take place by necroptosis. Yes, necroptosis associated to cell death will include 4 steatohepatitis, acute pancreatitis, reperfusion injury, neurodegenerative diseases such as Parkinson's disease. First is preoptosis. Preoptosis means preo means fever, apoptosis means optosis means apoptosis. Preoptosis is the release of the fever-inducing cytokine interleukin-1, which will be as the biochemical stimulates similaries with the apoptosis. Preoptosis occurs in the cells infected by microbial organisms. Infected cells will take place preoptosis, which causes the cell death. Autophagy means is a process cell eats their own contents. Caused by the delivery of the cytoplasmic lysosome only. In this condition cancer conditions, Alzheimer's disease condition, aging conditions, and the lysosome will start to eat their own cellular contents, and autophagy occurs. Ischemic reperfusion injury. Ischemia means reduced blood supply. In this reperfusion means if for a moderate period of time the blood supply is reduced. Next, the clot is removed, and the blood is going to the injured cells, and that is known as a reperfusion. Sudden flow of blood into the injured cells. In that, it will lead to an injury. Restoration of the blood flow causes recovery of the cells or reversible injured cells. But the restoration of the blood in the ischemia condition, it will further cause the injury. Rapid restoration of the blood in myocardial infarction can cause more myocardial injury. Yes. Why new tissue cells after restoration? Because increased generation of there will be in reperfused cells there will be more oxygen. So this more oxygen will combine with the electrons and form a reactive oxygen free radicals. This will further damage the membrane, DNA, protein, and intracellular calcium also. In that reperfused blood there will be more calcium. This calcium will get accumulated in the mitochondria, and mitochondria will not produce has less ATP only. So without more ATP it cannot efflux the calcium outside the cells. So intracellular calcium will accumulate and cause further damage. Reperfusion recruits circulating neutrophils and cause more neutrophils will come to that area, and It will cause inflammation and activation of the complement system. A free radical. Free radicals are chemical species which that they have a single unpaired electron in an outer orbit. These unpaired electrons are highly reactive and attack the proteins, lipids, carbohydrates, nucleic acids, and cause the cell damage and cell aging. Superoxide anion, hydrogen peroxide, nitric oxide, hydroxyl radical are some of the examples of the free radicals. Free radicals, reactive oxygen species of free radicals produced normally in the cells during mitochondrial respiration and energy generation. In that, increased production or decreased scavenging of reactive oxygen species may lead to excess of the free radical and condition called oxidative stress. Generation of the free radicals. The reduction oxidative reaction that occurs during normal metabolic process, absorption of the radiant energy, UV light, X-rays, produced in activated leukocytes during inflammation, enzymatic metabolism of exogenous chemicals of our drugs generate free radicals. Nitric oxide will generated by endothelial cells, macrophages act as free radical. Effect of the free radicals and the effect of the free radicals and other free radicals are cell injury, cell aging by lipid peroxidation in membrane. Oxidative modifications of proteins may damage the active sites of enzymes and generate misfolded proteins. Free radicals cause DNA damage. Remember how we can remove the free radicals. Free radicals are unstable and decay spontaneously. Antioxidants that block free radical formation are inactivate free radical. A series of enzymes that break down free radicals catalyze inactivate H2O2. Superoxide dismutase converts superoxide anion, glutathione peroxidase. Free radicals are chemical species that have a single unpaired electron in an outer orbit. Unpaired electrons are highly reactive and attack the proteins, lipids, carbohydrates, nucleic acid of the cells. Next, generation of the free radical. The reduction oxidative reaction that occurs during normal metabolic processes, absorption of the radiant energy, example, UV X-rays, produced in activated leukocytes during inflammation. Enzymatic metabolism of exogenous chemicals or drugs generate the free radicals. Free radicals are inherently unstable and decay spontaneously. Antioxidants, series of enzymes that breaks down free radicals, catalyze superoxide dismutase, glutathione peroxidase. Effects of free radicals on cells: lipid peroxidation in membranes, oxidative modifications of protein, lesions in DNA. Lesions in DNA, single or double strand breaks in DNA, cross linkage of the DNA, ultimately causing DNA damage. And these are the irreversible cell injury. In reversible cell injury, what we have seen is both we have seen necrosis. Necrosis means continuous uncontrolled cell death, and apoptosis we have seen. Apoptosis means programmed cell death. In necrosis, there will be a difference between necrosis and apoptosis. Means necrosis, there will be cell will get cell will get swollen and death, and in that there will be apoptosis, there will be shrinkage and death. Mostly neck there will be this how the irreversible cell injury occurs means due to the prolonged and severe cell injury only irreversible cell injury occurs. And first of all, in there will be decrease in the ATP, and next calcium will influx of calcium will lead to mitochondrial damage, and cell membrane will get damaged and lead to increased increased water into the intracellular components, and cell will get swollen and die. Okay, this all we have seen in this video. Thank you.